Hi, this is Dr. Collins Jones. Welcome to segment 8 of my video series on measurement, significant figures, and basic data analysis. In this segment, we will introduce ourselves to the concept of data and how to handle data. This is really meant to be just a quick overview. There are lots of excellent references and computer programs that can help you with this, but this is sort of just to give you a broad overview of the importance of data, what we can do with it, how we can present it. So let's start just by defining data. Up to this point, we've really been focused on with the idea of measurement and significant figures. We've really just focused on a single measurement, a single point. But really, when we go into the lab, we collect data. And data is a collection of many measurements, and these measurements are related. We're trying to find an answer to something. So preferably, as we collect our data, as we present our data, we're going to organize it into a table or a graph, and we're going to analyze it statistically. And in an upcoming segment, we'll give you just a brief overview of the very elementary descriptive statistics that you can apply to data, right? So data then should become information that's a record of the knowledge that we are discovering. So data describes a quality or a quantity, and raw data refers to the direct measurement or observation that is collected before any analysis or modification has been made to the data. So the raw data is the data that you read right off of the display on your spectrophotometer, on your balance, on your pH meter, that you measure with your meter stick, whatever. Before you do anything else, that's the initial data that you've taken down and put into a data table. Okay. Variables, you can kind of consider a variable as a container to hold and analyze the collected data. And then what we're going to try to do is use analysis, usually statistical analysis, to identify relationships between the different variables. So when we again look at what is data, it's measurements and numbers, it's observations, and we take those measurements, those numbers, those observations, and we summarize them in graphs and tables and charts to make it easier to try to understand how the different measurements that we make, different variables, are related to one another. So the types of variables. There's an independent variable, right? And so this is what's being deliberately changed. You control, if you will, the independent variable. You're changing the time. You're making a measurement every 10 seconds. You're changing the concentration, okay? So the independent variable is what we are deliberately changing, and it is plotted on the x-axis. And the dependent variable is what we are reading or what we are measuring on the instrument. And the dependent variable is changing because of the way that we changed the independent variable, right? So, and we plot the dependent variable on the y-axis. So, the faster way to say this is what you change, you plot on the x-axis. What you measure or read, you plot on the y-axis, okay? And so the goal here is by making enough measurements, we might be able to predict the dependent variable before we make the measurement. A controlled variable is kept constant. So sometimes we have several things that could be changing during our measurements, and the basic rule, at least for simple experiments, is to change one thing at a time. So, for example, if we're measuring the rate of reaction, we might allow the time to change or the concentration of the starting material to change, but we may keep the temperature constant. So we're controlling that variable. But in another set of experiments, we may keep the amount of starting material constant, but change the temperature. So the controlled variable is something that could be changed, but we're not changing it. We're keeping it the same. Now, there's a new science that we're not going to go into here, and I didn't put it on this presentation, but there's something called DOE, design of experiments. And with design of experiments, you can statistically design experiments where you have multiple variables changing at the same time, and the statistics allows you to interpret what's important, okay? And then in the panels over here on the right, these are just some nice pictures that I grabbed off of images, and it sort of illustrates, what's going on. But I kind of like the how to identify the dependent versus the independent variable down here. So you ask these questions. Is the variable manipulated or changed by the researchers? So if we are changing the variable, then it's the independent variable, right? If it's not being changed by the researchers, if it's what we're reading, then it's the dependent variable, right? Does this variable cause an outcome? Yes. If it causes something to change, then it's an independent variable. If it does not cause something to change, if we're reading it, it's the dependent variable. Okay. All right. So if you want to pause and read this again, that's fine. But we just wanted to sort of give you the concept of independent versus dependent versus controlled variable. Once you collect the data, you're going to need to analyze it. And this is called data analytics, right? And this is the whole now field of data science. And so before, when we did data science, it was really within each discipline. But now we're training people to do data analytics to become data scientists so they can be applicable to sort of any discipline. So data analytics is the process of analyzing raw data to be able to draw conclusions and make predictions, right? So types of data analysis: prescriptive analysis, descriptive analysis, inferential analysis, exploratory analysis, predictive analysis, or causal analysis, right? And so I could spend an hour explaining all of these, but these are just the different ways that people look at analyzing data. And if this interests you, I encourage you to look up each of these with a web search. Okay. What I'm more interested in, especially for our biotechnology students and probably chemistry and biology students, is how to present data. That's part of the data analysis. At least to me, that's part of the data analysis. It's organizing and presenting the information so it's easy to look for patterns. It's easy to look for relations between variables. It's easy for other people who haven't done the experiment to be able to look at the data and figure out what's going on. Okay. So data presentations help to illustrate relations between variables. They make it easier for other people to understand what's going on. So the three most common ways to present data are tables, graphs, which are also known as plots, and charts. So here I'm just sort of illustrating some of the key things that we want to think about if we create a table. So every table should have a brief descriptive title, right? And normally in the first column in the table, we put the X value, the independent variable, and in the subsequent columns, we put the measured variables. The table, all the data in the table should have the proper units and the proper number of significant figures. We could include the units in the column headings, okay? So this could be, time, and this could be moles of product right here, instead of having to write it into each individual entry. We like to have a caption that explains briefly how the data was obtained. Just a quick detail, something, whatever it was, enzyme and substrate were mixed, and every 5 seconds the amount of product was measured. And then we like the number of replicates. So replicates is how many times we repeated each data point. So, and that's also referred to as the n value, lowercase n. So n equals 3 means we did this in triplicate. N equals 5 means we repeated this 5 times. And then you might want to mention any subsequent data analysis. So did we calculate the standard deviation, or did we calculate the standard error of the mean? Did we use a t-test to compare the different data? So you would mention this. So this could be a paragraph. It shouldn't be real long. It shouldn't be like a whole page, but it should be enough that somebody could read the caption and figure out what you did. All right. So to graph or plot the data, right, we use graphing software. So obviously everyone who's going to be in science should know how to use Excel and generate graphs with that. But we also use an intermediate level statistics program, which I highly recommend, called GraphPad Prism. So this will do descriptive statistics, like calculate the average and the standard deviation, but it will also do comparative statistics, so you can calculate 2 different averages and compare them. It will also generate plots for you and do different kinds of regression analysis, okay? So if you're using a graphing program like Excel or GraphPad Prism or some other one, you're going to enter the values, right, in the columns and double check each entry. Although if you make a mistake, it'll be obvious when you go to plot this. Generate the graph and see how you want the plot to look. And so in these graphing programs, you get options. So maybe you just want to leave this as a scatter plot, okay? But first, when we generate the table, we're going to provide some information again. A brief descriptive title, and then the x-axis will be the name of the independent or the changing variable. The y-axis will be the name of the measured or read value. That's the dependent value, okay? And then again, we have a caption to briefly explain how the data was obtained. This is really the same concept as we talked about if you generate a table to show your data instead of a graph or a plot, okay? But we also want to talk about the statistics and any transformations. So we could plot the data just as a scatter plot. Just put a bunch of dots up there, right? That's it, all right? Or we could connect the dots by a line. Okay, and in some cases, especially if you're using a graphing program like Prism, what you might do is you plot the points, but the graphing program will then give you a best fit curve, a smooth line that connects the most number of points. Okay, all right. Now, a couple of other things here. If we have done replicates, even if we've just done duplicates, measured each value twice, or if we've done triplicates, measured it 3 times, we would like to be able to plot either the standard deviation, that is the plus or minus, or the standard error, the mean. And so these are shown as error bars, and this kind of shows the range, the plus or minus, for each data point. Okay, if you have to use several different conditions and you don't plot them all on the same point, you're going to use a different symbol for each data point. And nowadays, you also use a different color line, right? A different color curve for each of the different conditions. And so at the very end of the segment, I've put up some real world images that I took from scientific journals to just see how people do things. And so some are good and some are not so good. This slide just illustrates different types of charts and what kind of relationships they illustrate and a little bit about what they look like. And I could spend the next 20 minutes going over every column in this particular table, but really I just wanted you to see that there are lots of different ways to represent data, and this is the type of things that you study if you take a class in data science or data analysis. Is what are the different charts? What's the best chart to use given the data that you have? What will do the best job of presenting your data? So lots of different charts and some explanations here. If you want to pause the video, you could take some time to study this, but I just really wanted to illustrate the idea that there's lots of ways to illustrate your data. Okay, one of the other things that we do sometimes is we do data transformation. So in data transformation, a mathematical operation is performed on all the values in a data set. It could just be one variable. It could be the x variable, or it could be the y variable, or it could be both. But whatever you do to say the first x variable, you must do the same transformation for every other variable. Same thing if you transform one of the y variables, you need to transform all the other ones. So basically we do the transformations because it makes things easier to plot. It might do a better job of illustrating a relationship between the variables, okay? And so what you're really saying is if the data is not normally distributed, or if you're trying to compare different data sets which have different standard deviations, you might have to do a transformation. And in the old days, before computing power was readily available to everyone, people did manual transformations in order to try to get their data to plot as a straight line. So, back in the 1940s, 1930s, even the 1950s, before computers were readily available and there was software to plot your data, you might do transformations to try to turn your plot into a straight line. because the slope of a straight line is constant, and you can always calculate the slope and intercept and use that to then as a predictive tool. Okay, so examples. This is not comprehensive, but these are some of the more common transformations that one does with the data. One is reciprocal transformation, and I'm going to illustrate this on the next page. You might take the log, either the log to the base 10 or the natural log of the data value. You might square it. You could, I didn't put it here, but you could cube it. You might take the square root or the cube root, all right? Or you might multiply or divide all the data values by a common value to make them relative to a standard value. So you might say things are moving through the chromatography system, and I have one standard that I know always comes out at 10 minutes. So I'm going to divide everything by 10 minutes to make all my elution times relative to that one standard. That would be normalizing the data. Okay. And again, there's lots of different ways you can do this. The curve fitting programs actually do transformations to give you the best plot. Okay, and I just want to emphasize, every data point has to have the same transformation, right? At least in that particular column. Okay, so here are some transformation examples. In the column on the left-hand side, labeled raw data, this is the data that you collected, right? So if we did a reciprocal, if we took 1 over the raw data, here are the values that we get. If we do the log 10, right, take the log of each of these, here are the values that we get. If we square it, here are the values that we get. If we do the square root, here are the values that we get. And if we just divide everything by 20 centimeters to make it all relative to 20 centimeters, here are the values you get. And so a curve fitting program might actually do this, okay? And then they would see which of these plots the nicest. And it would say, okay, here are the different transformations. Here's the curve that arises from each transformation. Pick the one that you like best. As I promised you, here are some examples of published data tables and plots, all right? So the first one, we do have a title, right? And we're changing the dose. We're feeding rats a drug. In this case, it's DDT, which used to be used as an insecticide. So we're feeding the rats different doses of drugs, and then we're analyzing the rats on different days for the amount of a particular protein. So all they really said was the values are the mean, which is the average plus or minus the standard deviation, and we used 5 rats for each data point. But it really doesn't give us much more information. So to me, this is sort of a weak table. Here we have a plot where we've measured multiple different things. We're looking at the assay of a protein function for a protein that's called PTP4A3, and we're doing phosphatase assays. And basically the caption here is actually a conclusion, but it doesn't tell us, for example, where the protein came from, how much we used, how long the assay was carried out for, what the buffer is, blah, blah, blah. But we have done a transformation, right? So here we've done a log of the concentration instead of just plotting the concentration. And we're measuring the fluorescence intensity. And we've used different symbols for each condition. And we've plotted the curves in different colors. And we have the error bars present here, okay? So again, just to illustrate to you, if you look at something in a journal or in a publication and you see this, you kind of understand what's going on. And maybe now you can make a little bit of an assessment about how complete the information is for the table. And there probably should have been a title here, but the title is actually in the caption. But again, in both of these cases, I think the captions are lacking sufficient information. Next page, another table. And geez, I really couldn't find too many good tables here. But here we're looking at clones, that is cells that are producing the antibody IgM, right? And so each clone gets a label, and then we're looking at how much, which IgM was expressed, and then the titer, which is how much of it, okay? But again, we're not really saying what the cells were, how long they were grown for, how was the titer measured, was this, were the replicate measurements of the titer done, and so forth, okay? This table is a little bit more informative. Here we're looking at PER-C6 cells, and they're exposed to a vector, right? So some kind of DNA carrying the S1 spike coding domain. So this is some kind of COVID study, and they're measuring the production of the spike domain and binding. And so again, here we have the percentage of positive cells. We have the dye that's binding to c-Myc, and then we're looking at the SARS antibody. And again, this is a transformational log, and we're measuring an absorbance. So there's a little bit better information here. And then finally, we're looking at the production of a protein, right? In this case, actually a virus AAV. And we're comparing a 125 ml shake flask to a 3-liter bioreactor, right? And here it's saying how we measured the titers by qPCR. The data were normalized to the titer at 30 mils for the production scale. And you can see the error bars. And what you can't see here is, in real life, the error bar not only goes above, but it goes below. So these two are essentially, there's not much different in titer whether we're producing our AAV virus in a shake flask or in a 3-liter bioreactor. So just trying to illustrate what some real life tables and graphs and charts look like. And that is the end of segment 8.